Good morning, everyone. Welcome to Nutritional Biochemistry in the first semester for 2021. And we hope this semester is going to be crazily busy, but exciting as well and engaging. Okay, so this session is on macronutrient pharmacokinetics. Bit of a tongue twister, but we'll go through it. What's it about? But before that, I wanted to talk to you regarding the course structure for this subject. So I'm hoping you had the chance to look at the subject outline. So this subject is going to have 3 assessments. So first is going to be a short report in which through a case report, you'll be answering 3 questions. Second one is kind of little more intensive. That one will cover your session 5, 6, and 7. So we'll go through it. There are already videos there, FAQs there on LMS. I encourage you all to go through that. I'll also be having separate webinars for each assessment. So come to me with all your questions and we'll brainstorm through that. Another important aspect in this virtual environment of learning, which is fairly new from most of us, we have got kind of a lesson plan going. So there are some pre-lecture activities and post-lecture activities. The aim is that, in your biochemistry, chemistry, as well as foundations of human nutrition, you have studied some of the foundational knowledge required for this subject and your prospective clinical subjects. So I would like you to come prepared with that previous knowledge so that we go through this relatively new content in a more calm manner. For example, for this one, I'm hoping you would have gone through the cell membrane and how the nutrients are transferred across the cell membrane in our body. Then every week there would be some post-lecture activities. Target is to spend about 1 hour on both of these pre and post-lecture activities. Mostly I have planned them using H5P software. However, you would notice in subject outline, I might have written some other software such as Kahoot. But when I was learning to make these activities, I was really comfortable with H5P. So I ended up making the same activity. The name is same, it's just that software I used was different. So important thing, there are some quizzes. Now these quizzes would help you revise the content because over the years, I have coordinated this subject now for 5 or 6 semesters and have taught it for 7 or 8 semesters. So I had this feedback. Students used to feel that there is too much content and they can't retain it till the final exams. So these multiple choice question or single answer questions are designed so that you revise the content, weekly content, and some of the final exam questions. Please remember, not all the questions, but some of the exam questions would be similar or maybe the same to these quizzes. Okay, so stay with me and feel free to raise any questions. As this session is recorded, I encourage you to write your, any question that is coming to your mind and it is stopping you to concentrate on the session, to write it in the chat box. And I plan to go back to it in about 30 to 40 minutes, take a break, answer those questions, and then we come back to the session. These are 2 hour session, but we also have 15 minutes to 20 minutes at the end of the session. We can talk about any other questions in that time as well. I also like to take a break at about 1 hour. So that you and me, we both kind of take some rest, gather our thoughts, grab our teas or caffeine, and then come back. Any questions so far? Okay. So I request that you keep yourself muted, and when we are reaching that F&Q phase, you can switch yourself on back in. So let's dive into the details of first session. Here we are going to try and kind of revise what we know about digestion of macronutrients. As you would recall, carbohydrate, lipid, proteins, they are macronutrients. Then another important aspect of this session is that we are going to delve into understanding how these nutrients are absorbed in the intestinal epithelial cells. Okay. Another aspect or another important thing about first 4 session is, including this one, that we learn about other nutrients that help these macro or micronutrients to get absorbed or to get stored or to get transported. Learning this information would assist you in completing your first assessment as well. Plus, it will also give you some clinical tools for your prospective practice. Because, you know, there are times we get a client and we are looking at their biochemical report and we can see that they are deficient in, let's say, iron or B12. But when we compare it with the diet, we see they are consuming enough of that particular nutrient. So we kind of go into our investigative mode. And that's the time when we wear our nutritional biochemical glasses to see that maybe the nutrients that support this particular B12 or iron to get into the intestinal cell is missing. Okay. So that's what we will go through mainly in the first 4 sessions. So first topic for today's session is on transport mechanisms. And I'm assuming that you would have gone through the pre-lecture activities. And I encourage you to share with the group and tell me, what do we mean by active transport? Feel free to join in. So what is active transport?
So the chemical has to move against the concentration gradient.
Yes, thank you. Is that Jane?
Yes.
Thank you, Jane. That's correct. Anybody else would like to comment? That's fine. It's too early in the morning. I understand. Okay. Just one more thing. I have this nag or habit of adding new pictures. So when I'm making the lecture, I want to make it a little bit more interesting. So you might find that sometimes the slides from your PowerPoint slides, they are a little different. Let me know if it bothers you a lot and I'll stop doing that. Okay. So the thing is, this membrane, what we are talking about, this cell membrane is fascinating. Okay. It has hydrophilic lipophilic, that is water-loving end outside, and fat-loving tail inside. The idea is that it has to be selective to which nutrients it's want to get in and to get out. So it's kind of a gatekeeper for our body. Okay. So sometimes these substances, it can be calcium, sodium, potassium, even sugars and fatty acid. And sometimes it could be waste product. So this cell membrane is the one that decides that which molecule has to be moved out, that is against or up the concentration gradient, so that the equilibrium is broken. Sometimes there is an immune cell. Now, immune cell has to do a lot of work and it has to go against so many negative factors. So at that phase, we need active transport. And another important thing is that it uses energy because it is going to break that normal equilibrium of the cell. Okay. So just added something here, that it transports molecules through the cell membrane against the concentration gradient. So more of the substance is inside the cell, that is a nutrient, or outside the cell, that is a waste product. Then we come to the passive transport, which is the reverse, because it is the movement from an area of high concentration to an area of low concentration. And this transport mechanism includes simple diffusion or facilitated diffusion. So the whole idea is that passive transport is trying to make the equilibrium. And because it is the normal way for a cell or a body to do, it does not require extra energy. Okay. And what is the difference between diffusion and facilitated diffusion? Would anyone like to comment here? Okay. So facilitated diffusion is, you can see these different nutrients of different green color, they can move across the cell membrane. However, facilitated diffusion is that there is a channel and then there is a carrier, which can be a protein also, protein carrier. It kinds of hold the hand of this nutrient and carries it across the membrane. Okay. So again, kind of some friendship going on between the carriers and between the nutrients as well as the cell membrane. That's how I like to see the role of different nutrients in our body. They help each other out. So again, kind of reiterating that passive transport maintains dynamic equilibrium of water, gases, nutrients, waste, etc. between the cells and the extracellular fluid. So we will be discussing different transport mechanism for different macro and micronutrients. Okay. Now there is a word here, pharmacokinetics. What does that mean? So it is actually derived from the use of drugs. Okay, that's why it has that pharma attached there. So it actually means what does the body do to that drug or a nutrient after we consume it. That means we absorb it, then we distribute or transport it in the blood, then we metabolize it in the liver as per the requirement of the body. And if its end product are toxic, then we secrete it via liver and then through kidney, or exhale it. If it's carbon dioxide, the end product, we will exhale it through lungs. So that's what we mean by macronutrient pharmacokinetics. So we will be going through how carbohydrate, fat, proteins are absorbed, then transported, and maybe if we need to go further about how they are excreted. Now, this entire process can be impacted by various processes, and I'm sure you would have example of each one. First one is pretty simple. If somebody has a poor digestive function, that means the enzymes required to break down the food bonds is not working properly, so we cannot get it inside our intestine. What do we mean by mucosal integrity? Do we have an example here how this can impact absorption of any nutrient? Feel free to comment here. Okay, I've again added a picture here and I'll spend just few seconds here. When we mean by mucosal integrity, we are talking about the intestinal cell. And as you would recall from your biochemistry that the small intestine, it is actually made up of a layer of single epithelial cells. And this picture represents those epithelial cells. Towards the lumen, that is from where the digested food is coming, that is called as brush border. So that's the first stage of absorption of a nutrient. This is called brush border, and we have mucus lining this brush border. The thicker the mucus, the more stronger our brush border is. And why does it has to be stronger? Because it wants to filter. You know, mucus is thick, so it will stick to any bacteria or any bacterial material that we might have consumed or which might have passed through the strong hydrochloric acid. Okay. Then the foods or the nutrients, through active or passive transport, they enter the brush border and they reach inside the cell. Sometimes body stores some of the nutrients in these cells. Sometimes it transforms it to a different product. Why? Because it wants it to reach the blood. And how does it happen? So that's the second phase of that absorption, which happens at this second layer of the epithelial cell, and it is called as basal membrane. Attached to this basal membrane is our blood vessel, okay, or arteries. So the nutrients, they travel from here, brush border, and then travel through this intestinal cell body, and then via basal membrane, they reach the blood. So throughout first 4 sessions, we would also be learning about the transport mechanism, active, passive, carrier protein that are present at this basal membrane. Okay. So when we mean by mucosal integrity, we are talking about this area. Okay. So sometime if somebody has a very thin layer, that means they are susceptible to attack by any toxin or bacteria that may be reaching there. Then intestinal microbiota. Now, this is a fascinating new word for all practitioners, and we have a separate session for that. Actually, if we have good bacteria, healthy bacteria that are residing in the colon, they kind of secrete such compounds that support this brush border. So it is stronger. Okay, and our intestinal cells are happy. And we'll talk about it in detail in coming up sessions. Then agonist and antagonist, how does that impact any nutrient absorption? Any example? Or maybe example for dietary fiber? If I'm consuming too much fiber, what happens to micronutrients such as iron or calcium?
They compete to get into the cell?
They prevent it, yes.
Yeah.
So you're on the right track, Megan. So what happens is dietary fiber binds to iron and calcium. It makes insoluble complexes. And everything is excreted rather than going inside the cell. So well done. Thank you. What about agonist and antagonist? How would that affect? Or any example? And no question is wrong. No answer is wrong. We are just trying to understand some new things, and it's normal that we sometimes speculate. Have you heard that they say that vitamin C supports iron absorption? Have orange juice. Okay. So sometime, again, we will explore this in the coming up session. Remember, I was mentioning that there are transporters here and here. So sometimes these nutrients, they carry or move around with the help of other nutrients. So they become agonist. Vitamin C is one such nutrient that supports iron absorption because it converts it into a form which our brush border of the intestinal cell love. Okay. Whereas, as we just mentioned about dietary fiber, it becomes antagonist to iron. Similarly, calcium. Calcium also kind of binds to iron and makes it insoluble. You know, sometime a practitioner or a GP would tell people to have iron capsule and don't tell them how to consume it, and people take it with milk or tea. And tannins in milk, tannins in tea and milk has calcium. They both are antagonists of iron. Okay, so these are very small things, but they matter a lot for micronutrients which are required in small amounts in our body. And then stress. Who is stressed today for starting nutritional biochemistry?
Me. I didn't sleep.
No one.
Definitely.
Well, the whole idea is not to be stressed, okay? That's why we have a full chapter of one session on stress and HPA dysregulation. Because little bit of stress is good. However, when it is chronic or long term, it harms the microbiota and it also disintegrates this brush border. Okay. So remember that when that stress kicks in, what do you do to get rid of that stress? We take deep breathing, we listen to music, go for a nature walk, sit in sunshine. It all helps. Okay, and these are the lifestyle intervention that we'll be telling our clients in future. So best to apply it all on ourselves. Okay. Okay. Any question or comments? Like my flow? Am I too slow? Am I too fast? Let me know. I'm happy with all kind of feedback. Okay, so I've got a green light that I'm doing okay in the chat box. So let's move on. So we are going to start with carbohydrates. And I'll kind of rush through first couple of slides because you have studied it in various subjects. But still, if you have a question, let me know. So we all know carbohydrates. They are made up of carbon, hydrogen, oxygen. We have different types. Monosaccharides, which have one glucose group, whereas disaccharides, which are made up of two monosaccharides. So the most abundant one, monosaccharide, fructose, galactose, glucose. Then we have disaccharides such as sucrose, maltose, and oligosaccharides, which are short chain monosaccharides, about 3 to 6 units. And then we have polysaccharides that cannot be broken down in our intestine by any enzyme. However, we have got our symbiotic friends, microbiota or bacteria, which are there in the gut that act on it and do amazing wonders and support us. So when we are absorbing this carbohydrate, their absorption will decide what's going on, what's reaching the bloodstream. Enterocytes. So remember, we will be calling these intestinal cells enterocytes. So entero means inside and cytes means cell. Or we'll be calling them epithelial cells because that's what they are made up of structurally. So mostly these intestinal cells, they absorb monosaccharides. Sometimes very few disaccharides may be absorbed. So what does that mean? Before reaching that brush border, we have to break down any disaccharide that we may have consumed. And this process is done by glycosidases. So there are some linkages between two monosaccharides, and these enzymes, they break that bond. Now, digestion of disaccharides, it will occur at the level of brush border. And these enzymes are known as disaccharidases. I've already shown you this diagram. Even if when you are doing self-study, you will notice that this diagram is presented in various ways. I'll again mention that there is a brush border here where the first step of absorption happens, and then there is a basolateral membrane which is next to the blood flow, blood vessel or artery, where the second stage of intestinal absorption take place. So we all recall that digestion actually begins in the mouth, where we have amylase enzyme. Then we have pancreatic amylase in the duodenum, and followed by amylose and amylopectin that are breaking down the oligosaccharide. So the main aim is that before reaching that brush border, we have monosaccharides so that intestinal cell can absorb them. This is just a summary from your previous knowledge, and I just have one interesting fact in my notes here, that the absorptive capacity of human intestine is about 5,400 grams per day for glucose. Okay, so that's what the experiments have shown, that we have the capacity to absorb too much glucose. And again, it might have been the evolutionary process that digestion of carbohydrate is so efficient, whatever monosaccharides we are consuming, they will be absorbed. Okay, so an important thing now in coming up slides about glucose transport. So now we have this glucose which is required by our body, but actually it is required by each cell for its energy because we are making ATPs out of it. But its concentration in the blood must be precisely controlled. Now, highly polar glucose molecules, they cannot move across the cell membrane by simple diffusion because it cannot pass through the non-polar matrix of lipid layer. So remember that inside the cell membrane is the fatty layer and outside is the water-loving layer, hydrophilic and hydrophobic. Therefore, for glucose to move around inside the cell and go across, we need energy-dependent transport, and they are called as sodium glucose transporter. Why? Because for an exchange of sodium, glucose is transported. So just highlight this in your notes. These are the kind of questions that might be asked in the multiple choice questions in the exam. And I like to give such hints during my lectures. Now, this was in the intestinal cell, but as we all know that every cell needs glucose. Okay? So some other cells, they may use carrier proteins to transport glucose. And they collectively are called as GLUT or glucose transporters. And you know, there are so many, we are discovering them even now. So far, you don't have to remember all of them. There are few, and that slide is coming up. So far we have identified about 14 glucose transporters. Now, as you would recall, what do these transporters do? They get attached to that molecule. They may undergo a conformational change so that the molecule is passed along the cell membrane and goes inside. So this is kind of a list. And again, you would notice I have highlighted a couple of them in red. So it will be a good idea to save it in your notes. So you can see how much variety do we have. Each cell has got a separate GLUT transporter. Each cell needs glucose. That's why after this lecture, I'm going to go for some chocolate or a mocha to get those brain cells some glucose. Okay, so just a list to show a variety of glucose transporters. And then this slide is again a summary of carbohydrate absorption from your previous knowledge. Don't want to repeat all that unless you have a question. Now coming on to the absorption of other monosaccharides such as galactose. Now they are absorbed in the small intestine by sodium glucose symporter or sodium glucose transporters. At high concentration, that means if I took, let's say, carrot cake with my coffee in the morning, then all this sugar will be transported by GLUT2. So body can shift. Body is prepared to take up all the sugar. It is conditioned, I think. Then glucose, galactose, and fructose all exit the enteroside via GLUT2. So for entering inside the cell, we have different mechanism. However, for exiting at the basolateral membrane, we have only GLUT2. Again, I encourage you that for the first 4 session, you may make yourself a kind of a table, because if you end up revising it at the end of session 13, there might be some confusion. But if you're making your notes, transport mechanism for glucose at brush border, at enteroside, transporter protein in blood. So with such subheadings, then before the exam, you will have a summary. You won't have to go through all the lectures. You will have your summary to revise that content. Okay, moving on to fructose. For fructose, we have got GLUT5. So we have specific transporters, but rate of uptake is slower than that of glucose and galactose. However, it is increased when GLUT2 is present. It exits enterosides via GLUT2. And we all know what happens. It goes to the liver, it is rapidly phosphorylated, and then it is available for all other work. In fact, excess fructose is converted to fat as well. GLUT2 again for facilitated transport at basolateral layer for glucose and galactose. And then we have specific transporters in the skeletal muscle, heart, and adipose tissue, which was mentioned in that diagram in that table, couple of slides below. But important thing to remember is that we need few nutrients to support insulin receptor function. And these are chromium, glutamine, and vitamin B3. So I encourage you to remember that. And how and where does that happen? So I assume you would have seen this image in any of your biochemistry subjects. So every cell wall, we have these insulin receptors. Okay? Whenever there is sugar in the blood, we all know from the beta cells of Langerhans in the pancreas, our body secretes insulin. So when this insulin comes outside cell membrane, it attaches to its receptors. And now this receptor, remember I talk about conformational changes, it changed shape. Okay. This phosphate molecule at the end, it is kind of a signal. And then through this signaling of MAP and PI3K, for this course, you don't have to remember the full form, but if some of you want, I can talk about it. So what happened is that insulin signal gives message to the glut, which is lying inside the cell. Hey, buddy, glucose is outside. Move out. You have to do some work. So it is through this signaling that glut now knows that it is time for his work to start. So it moves outside or on the cell membrane. Now it has a slide here. So glucose comes inside. And the latest understanding in insulin resistance and diabetes type 2 is, we all know that in type 2 insulin is being made. There is a glut transporter, there is an insulin and insulin receptor, but then what's going on? Why is glucose not going inside the cell? Our latest understanding is that there are disintegration or some lost signaling here. Okay, and chromium, niacin, glutamine, they all support these signaling. In fact, chromium, as we will study in session 4, can potentiate insulin receptor function. Cool, isn't it? It's so fascinating. Okay, time to take some break. So we have a question here from Courtney that could someone who is vitamin B deficient then possibly have issues with insulin? Would somebody like to say something? So as we mentioned in a couple of slides ago, that insulin receptor needs vitamin B3. Okay. For some reason, if somebody is not consuming B3 or is consuming in a way that it is not available to the body, and we will explore about this in next session. Yes, definitely. That's what our aim is, to learn through this session how there is kind of a friendship going on, a big happy family of these nutrients that help each other. And sometimes, like in our family or with our loved ones, we have a heated discussion. Similarly, sometimes these nutrients, they compete with each other as well. So yeah, Courtney, so you are right. It is possible. Okay, cool. Any other question? Cool. Shall we continue with 5 minutes break? Now we move on to lipids. Again, just trying to refresh your previous memory that we have different kinds of lipids, fats, oil, or waxes. Mainly what we are consuming via dietary fats are triglycerides, and for some reason now they are called as triacylglycerols, so TAGs. So in the slides and other sessions, we may be using TGs as the abbreviation or TAG. So it is the one and the same thing. Different type of lipids in our body: fatty acid, TGs, phospholipids, sterols, etc. And we know how important they are. They are not only fuel source for our body, but they regulate icosanoid synthesis. Has anybody heard this name before? So this icosanoids, again, we have the entire, one of the session 10 is entirely on icosanoids. So these are inflammatory molecules, okay? And they are derived from fatty acids in the cell membrane. So we'll delve deep into that. What are some of the other functions of fat? Would somebody like to comment? Or any signs or symptoms of deficiency of essential fats that you may notice in any person? Very good, Megan. It is required for brain function. And yes, it is important for giving warmth to our body. Anybody else? Apart from being tasty, adding goodness to our food, skin integrity, isn't it? Sometimes people have dry skin and flaky skin. Yes, Jane is right. They are required to make hormones. Yeah, good. Yes, Stephanie, correct, inflammation. And as I mentioned, we'll talk about that because there are two aspects of inflammation. So yes, it is via fats that we generate inflammatory molecules that are eicosanoids. Very good. And thank you. And we remember there can be fats can be saturated, monounsaturated, or polyunsaturated. Summary of lipid digestion. We have these enzymes called as lipases, which break down the bigger chains of fats into fatty acid and glycerol molecules. Then we need bile because it emulsifies lipids, and we'll delve deeper in the coming slide. Then we have pancreatic lipase and so on. So now these fats, they are hydrophobic. And all the enzymes, they are water loving. So this digestion is special. And what we call it as the emulsification process. And there's an important role of bile salts in this. So the idea is that there is this video, it's a 10 minutes video. Either you can all watch it on your own because owing to copyright issues, I can't play it in this live presentation. And then we come back and discuss its component, or I go through the slides first and then you go and look at this video, whatever you are comfortable with. Tell me, you want to watch the video first or go through the slides first? Okay, one vote for looking. Okay, okay, let's go through the slide. Great, great. Let me try and okay. I'm going to make some drawing and I'm not very good at it, but it makes sense and the slides would be much easier later. Okay. So this is my version of epithelial cell. Okay, apologies, it's not beautiful. So now what's happening if I had some food which had a lot of fat, okay? Let's say it is a carrot cake. Now this fat, as we know, they are long molecules, lot of chains. Okay? Now, but body cannot absorb it because remember that this is fat loving, whereas this layer is water loving. Also, lipase has to act on it. But how would lipase act on it? Because it is also, it likes aqueous medium. So what happens is that bile salts come into play. As soon as fat is absorbed, body sense it, and pancreas release these, gallbladder release bile salts. Now, what do these bile salts do? You know, they are like detergents. When we are working with some some fat, let's say olive oil, kneading a pizza dough or something, we have oil, and if we wash it with a soap, it takes away the grease. So what these bile salts do, they kind of go and break this fat from the food into small molecules. And remember, bile salts are unique. They have both water-loving end and fat-loving end. So the way they make small droplet of fat is that their water-loving end is facing outside and fat loving end is binding to that small globule of fat. And then what they make is, it is called as a micelle. Now this micelle reaches the brush border, and because now this fatty chain is kind of smaller molecules, lipase can attack it at brush border and breaks it down into fatty acids. So it separates that glycerol chain from the fat chain. And the fat then simply goes in by diffusion, because it's kind of that molecule which has water-loving chain outside. So these micelles, they just travel inside. Okay. So now let's go through the slides which have the same thing written, that triglyceride molecules, they first must be enzymatically digested to yield monoglycerides and fatty acids, so that they can simply diffuse inside the intestinal cell. And this is done with the help of emulsification, which is carried out by bile acids. So on exposure to large aggregate of TAGs, the hydrophobic portion of bile acid, it kind of intercalate into the lipid with hydrophilic domains remaining at the surface. So this is a better drawing than what I did. So now these are small droplets of fat. Now these droplets are ready. So we are in the intestine, and the lumen, the liquid area outside the intestinal cell, has got pancreated lipases. They now act here and separate fatty acid from the glycerol chain. The free fatty acid, it remains bound to the bile acid to form a structure called as micelle or micelle. And these micelles now simply diffuse into the brush border. So this is the first phase. Any question? Sorry, can you say it again? Good, Jane, thank you. No comments, no questions so far? Easy peasy.
Fine.
Cool. Now let's come to the second step. So these micelle particles, they are water soluble to penetrate the enterocytes in the small intestine. Now, crazy but fascinating thing about our body is that at this intestinal layer, it breaks down everything to its basic form. However, our body uses nutrients in its natural form. Okay, so in the intestine, body again tries to reform it to its natural form. And this stands true for all other nutrients as well. So when it is getting confusing, remember, body tries to go back to the natural form. So inside the enterocytes, there is re-esterification and reformulation of triglycerides happening. Okay. Now, when it does that again, the problem remains the same. How does it cross the basolateral layer now? Okay. So now what body does it, that so inside the intestinal cell, body takes the fatty acid and cover it by lipoproteins. Okay, again, these molecules of lipoprotein would have water loving end outside and fat loving portion inside. And what the structure that we make here is called as chylomicrons. Can't write with this, sorry. Okay. Okay. Now this chylomicron, it leaves the basolateral layer. But these are really big molecules. It has fat inside and then protein outside. And our arteries next to the basolateral layer are quite thin. So these big molecules cannot enter the blood actually. It enters the lymphatic system. So these chylomicrons, they leave the basolateral layer through lacteals and travel via lymph system. Okay, so that's a fascinating thing. And all your fat soluble vitamins, they will go with this. Okay. But if there are short chain fatty acids, then they kind of attach to albumin and then they can freely move around in the blood. But when we are talking about the little bigger fatty acid, they have to go as chylomicrons. This is the major difference with the any other molecule which is being transported via basolateral layer. All other will enter the blood, but fats, they would enter the lymph. So lipids, sorry, can I request you all to mute yourself? Thank you. Now lipids, they are transported as lipoprotein, and there is a whole new word about new information coming out of these lipoproteins. So these lipoproteins, they differ according to the ratio of lipid to protein that is in that body. And that protein portion is known as apolipoprotein. And it is relevant because there is research that sometimes people with familial hyperlipidemia, they might have genetic issues of making a particular apolipoprotein. So the chylomicrons, because it is being made in the intestinal cell where all the fat is being accumulated, they are the most dense. So they have the maximum amount of triglyceride content attached to them. Then we have the VLDL, very low density lipoprotein, intermediate density lipoprotein, and I'm sure you are familiar with LDL and HDL. So one of the reasons that HDL is called a good fat is because it has the highest amount of protein attached to it and it has limited amount of fat. And there is consistent clinical data that high HDL level is associated with lowered risk of CVD. And we also know that it can be modulated by exercise and yoga, reducing consumption of trans fatty acid and increasing omega-3 and soluble fiber. As you would recall, HDL is the one that is carrying fat away from the organs to get metabolized. So remember I was talking about apolipoprotein. So if there are different type of apolipoproteins, great number A, B, C, and D. But I would like to draw your attention only to two. So one is that when there is deficiency of these apolipoprotein A1, it is associated with HDL deficiency. So Tangier disease is one such disease where there is deficiency of HDL. And as the second word in this sentence, amyloidosis, it's making of amyloid concentrate. And it is associated with Alzheimer's disease. So some people may genetically be making low ApoA1, and hence they may have a tendency to make these amyloids that may appear in brain, as in case of Alzheimer's. So it is kind of a relatively new research. And then ApoB is the another one. There is data that if there is high levels of apolipoprotein B, then it is related to heart disease. So sometime if you have a client coming to you whose diet seems okay, that person may be working out a lot, but still you would notice that their LDL is quite high. And if you go through their family history and you realize that they have a familial hypercholesterolemia, it will be good idea to get their apolipoprotein B checked, because we can check it. When we do lipid profile, we can ask them to check ApoB, and it will confirm your clinical speculation and diagnosis that it may be a genetic issue. Okay, I know there's some discussion going on the time. Yeah, I thought the time was AEST time, and that's what I was given separately and you were given separately. So no issue, we have a session in the afternoon, and this session is recorded, so you can visit it again. And we have 3 other sessions, Thursday morning, afternoon, and Tuesday also. So that's why we have a lot of options for you people. You can log on to any session, but only this session would be recorded. So you can visit that anytime till the end of semester. Okay, any questions regarding absorption of lipids? Good. We are moving along fine. Okay. Feel free to go through the session later in your own time. And if you have any question, post it on Loops. Let's move to proteins now. Proteins are the principal nitrogenous constituents. We all know that they make our skeleton muscles, everything actually. And the only difference is that they have a amino group, and that's how it differs from other macronutrients. And their distinctive characteristic is that they have amino acid side chains that make up polypeptides. That is how a protein is made. So there are, it can be a tertiary structure, which is kind of very long, and it might have dipeptide and then tripeptide. And we all would recall that we have some amino acids that are essential to us, that is, our body cannot make it, so we have to consume them through diet. And then there are some non-essential, which the body can make from the carbon skeleton. Remember your Krebs cycle, 3-carbon tricarboxylic cycle, where we are making all these amino acid interchangeably. And there are some conditional essential amino acid, that is, they are usually not essential except in the time of illness and stress. There are few of them, and if anybody remember it now, I would love to hear it. Otherwise, this is your search query for today. Do your own search and tell us on loop which one are the conditional essential amino acids. Now, structure of proteins can be studied in different forms. It can be on the basis of type of peptide bonds, or it can be on the basis of their chemical structure, whether they are acidic or alkaline. So peptide is the family of molecules formed from linking of various amino acids in a defined order. And the link between one amino acid and the next is an peptide or an amide bond. And polypeptide would be kind of a small proteins, which would have about 10 to hundreds of amino acid attached to it. And this is one such example. So you can see that there is a chain of different amino acid in a particular sequence. And that is the particular sequence that is coming from your messenger RNA via transcription from your genes. Okay. Any wild guesses which hormone it is? Any guess? Because obviously you are not supposed. All right, thank you, Megan. Megan is absolutely right. This is insulin. How did you know? Good, good. Yeah, so this is insulin. So you can see that it's an intricate structure. And that's how we make all the hormones or any other thing in our body. Chains of different amino acids in a particular order. And body knows which one to go and after which. Okay. Absorption, we all remember from our previous knowledge that digestion begins in the stomach. And the gastric cells, they release hormone gastrin, which enters the blood causing release of gastric juices. And hydrochloric acid in the gastric juice is very important because it denatures proteins and also converts pepsinogen to pepsin, so that pepsin can act on those protein peptide linkages and break down it into amino acids. Okay, so that's a very important step. Sometimes you may have people who will complain that after consuming a protein-rich diet, which could be a meat, or after consuming lentils, they are having flatulence or they are having undigested stools. One of the reasons could be that they have low hydrochloric acid, whereas there could be other reasons as well. Now, absorption of amino acids takes place throughout the small intestine. However, mostly they are absorbed in the upper intestine, small intestine. Again, they are very important molecules. So body expends energy, body uses energy so that amino acids can pass through the intestinal brush border. Mostly these energy dependent systems, they also require sodium. So if somebody has issues with sodium levels, then that will impact absorption of amino acids also. So what this diagram is depicting is that all after this hydrolytic digestion, tripeptide, peptide, and amino acids, they can be absorbed intact across the intestinal mucosa and then into the blood. So there are mainly two dominant transport systems. So one is a peptide transporters, and it has a high affinity for di and tripeptides. And these peptides, they transfer, these transporters, they transfer any di or tripeptides at the brush border. Then we also have specific amino acid transporters which use sodium. So when will that come into play? If I'm having that high protein shake in the morning, or if I am coming from a gym and I'm consuming a load of particular amino acid, then this system would be overused and it will need sodium. And it is really active process. Body uses lot of energy. Sometimes, body also differentiate between amino acid and alkaline amino acids. Basolateral layer is more permeable to the amino acids. So diffusion is the main process here for transferring amino acid. And how does that affinity, which I was talking about, happens? Branch chain amino acids are absorbed faster than smaller amino acid. What does this you must be having some questions in your mind when people are doing a lot of physical activity and they are consuming a particular amino acid, what's happening at the absorption level? So Megan is asking one question, and let's discuss about it a little. That is the specific amino acid transporter for protein shake? Is it because of the amount of protein? Good question, Megan. Tell me, what do you think? So what's happening is there are two things happening here. When we are consuming protein shakes, and if you look at their label, mostly they are made up of individual amino acids. So we are not consuming it as a food. So if I'm consuming a chicken, it is coming in the form of a protein. Body breaks it down, spends some energy in transferring and converting into amino acid, and then they go inside our body through using both these transporters, because some of those proteins may be broken down, some may not be. So body will be using both the transporters. However, what's happening with these protein shakes, we are consuming high amount of a particular amino acid, which may have all the essentials, or they come in different combinations. We are all aware. So body goes into a mode of a competitive inhibition. As I just mentioned, that body may have, you can see that there is a particular affinity. Branch chain amino acid are absorbed faster than neutral amino acids. Okay, so the body goes into a fighting mode. It has to decide which one to take inside. And in that phase, some of the amino acid may be lost. Okay. Did I answer you? Does anybody has any other comment on it? Aha. First, let's stay with your first question, Megan. Does anybody has anything to add to it? Did it make sense? Okay, now we have another important question coming up from Jane. Megan, was that first thing clear about difference between different amino acids? Okay, great, thanks. Now let's move on to your next question. Is this why protein bars, shakes can cause flatulence, bloating, etc.? Would anybody like to comment on that? Again, that's a very good question. There are a couple of things that may be happening there, Megan. One would be that person may have low hydrochloric acid. That may be causing an issue. And these bars are usually, they are usually keto bars, isn't it? They are high in protein and they are high in fat also. And if people, they don't have a strong epithelial layer, that can be one reason they are having bloating. And yes, third reason could be this, that there is a competitive inhibition going on and all the protein cannot go inside, it is staying in the lumen and it is causing flatulence and bloating. So there can be more than one reason and about this. Okay. Good. Now let's go to Jane's question. Jane is asking, can increasing sodium increase amino acid intake? Very good question, Jane. But I would like you all to comment on it. What do you think? Or even I can say, if somebody has a deficiency of sodium, would their amino acid absorption be compromised? What do you say? Yes, Courtney, you are right. Yes, thank you. Yes, because for amino acid to transfer, we need sodium symporter. So if somebody has worked out, they had sweated a lot, and they are taking a high amino acid shake with no sodium in it, then it can be an issue. And maybe you would notice that most of these protein shakes have high amount of sodium. Okay, and that high amount of sodium would actually impact, it can impact calcium and it can also impact potassium. And we would explore this more later. So the thing is, the body is always trying to be in a situation of homeostasis. And what we are doing is, we are playing with our environment, we are playing and stretching our bodies, and we are taking away from homeostasis. So our bodies have to do much more. That's my understanding. But you may correct me if I was wrong. Now there's another question from Tracy, that she's saying, I've also heard that liver can be overloaded with high protein amino acid consumption, causing the liver function to be compromised. Again, a good question. Would anybody like to comment on that? Yes, I assume so. You are right. It is actually not assumption, it is a factual thing, because body makes everything from amino acid, enzyme, hormone, storage protein. Iron is stored as a ferritin, vitamin A is stored as stellate cells in the liver, everything is protein. And excess proteins would overwhelm liver. Okay. Yes. So liver has many things to do. It is a factory. It has not only to do manufacture, but it also has to excrete toxins. So yes, it does get overloaded. And the other things that happen is that a lot of amino acid, it plays with the pH of our body and the kidney. Because when we are consuming a lot of meat proteins, we are sending a particular amount of amino acids and its byproducts to the kidney. And it plays with the hydrogen ion concentration, because all the enzyme in our body works at a particular pH. And this is an issue that we would be discussing in our last session. Okay. Yes, Jane, you are right. It will be increasing nitrogen levels. Yes, it will be increasing the pH that we don't want. Very good. Yes, it is interesting. Cool, cool, good. Okay. Couple of slides more. That transport of amino acid across the basolateral membrane is mainly via sodium independent transport. So at the brush border, we need sodium, we need energy, but at the basolateral, our body is more receptive and it is a trouble-free transport here. Sodium dependent pathways can still be used if the body feels that I am lacking amino acid. Concentration in that flow is low, body is intuitive, and body will translocate sodium ions to get more amino acid inside. And then they all go to the liver, where there are various mechanisms, because liver is the main site of its metabolism. So it uses more than one system. Okay. And we all know how important are proteins, and we used to make it all those different tissues, organs, and everything in our body. That's all for today. Any question, any comment? So we still have time. What do you think that I break you into breakout room and you can kind of spend, 10, 15 minutes knowing each other, kind of, only if you want. Courtney is asking if we have a new reading to revise. See, first 4 chapters are made from your main textbook, Gropper and Smith. Okay, so this is the one, right? So go through that chapter and you will find whatever we discussed today there. No need to refer to any other article for this one. For coming up 2 sessions, there are few readings because we will be now touching on all the water-soluble vitamins, all the fat-soluble vitamins. So I have put some pre-lecture reading material on the LMS because I want you all to be aware about the good food sources and deficiency symptoms of these nutrients. So go through that and then make your own table as well so that you can use it when you go to the clinic. Okay. For sessions after lesson 5, those lectures have been made using research articles because most of the information cannot be found in one book as such. So for them, there is a long list of references at the end of the session. And plus, if there is an important topic or any controversial topic that we need to go deep into discussion, then I will share some articles which you can go through in your free time. Okay, again, there is some issues regarding the times. So this session starts at 8 a.m. Brisbane time. So what would that time be as per Perth session? Okay, what we were told, yeah, this daylight saving can be confusing, isn't it? And it's going to change after 5 weeks. So after 4th April, you would notice that your links might remain same, but the times may change. So, okay, Tracy, you logged in 8 a.m. Brisbane time and it was late by an hour. Okay, yeah. So we were given, sorry, okay, we were given AEST time, Australian Eastern Standard Time. That is 8 a.m. And I think Brisbane time was 9 a.m. Yeah, sorry. So yesterday evening, some of you were texting me. I spent some of the time looking at time myself. So it will come to us, and the lucky thing is that this session is recorded, and you have 1, 2, 4 more sessions to log into, and then by next week you will have a good understanding which suits you best. So feel free to join again if you want. Yes, Talia, week 1 to 5 Australian Eastern Time. Yeah. Okay. We should have some table about different times. No, Tracy, attendance is not compulsory in any of the lectures. I am not recording it. Okay. We are, yeah, feel free to come anytime, join in, and yeah. And I work full time, and I like to get back to the students' query within a day time. So if you have any questions anytime, post it on the loop. And in week 4, I'll be doing a webinar for the first assessment. It is based on our session 2 and session 4. So it will be a good idea to go through that before we meet for webinar Q&A. Okay. Well, thanks for your lovely session. Hope you have a good day and a good week. Thank you, everyone. And let's leave it here. Bye.